thank you so much for joining us today. We're really happy to have you. Hopefully you'll get something out of it as well. So my name is Eva and I am one of the members of the iGEM team at Dundee University. So first of all, a little explanation, what is iGEM? So iGEM is a synthetic biology competition in which teams from all over the world take part to develop new solutions to old problems. And this project takes 10 weeks. And we worked in the molecular microbiology lab. And here's Kyle working away in UV light. And synthetic biology is a new area of study which combines science, all areas of science really into one. So it really takes into account engineering, mathematics, and all these disciplines which never really connected beforehand. And here's our team. So here's us. So there's 10 of us. We've got 6 biologists, 2 mathematicians, a software developer, and a physicist, sorry. And we've got 2 advisors, Richard and Lucia, who are PhD students, and we've got 3 supervisors, Professors Tracy Palmer and Frank Sargent, and Dr. Fordyce Davidson. So first of all, let's talk about algal blooms, because that's why we're here. So algal blooms are caused by the rapid growth of algae in water. So when there's a lot of nutrients, when there's a lot of light, there's a huge expansion in growth. And it's an international problem. And as you can see in Beijing Olympics in 2008, there was a huge algal bloom problem and 10,000 people had to be employed to clean up the algae just before the Olympic regatta. And it's also a problem here in Dundee. We've got a Swanee Pond here, and as you can see, we've got a public warning here as well. So let's focus on Clatto. Thanks to George, we have some wonderful pictures of the olden days, where Clatto is used for recreational activities. There's a lot of kayaking, water sports, and other activities. Unfortunately, it has been closed for over 6 years now, which decreased the numbers of visitors by 60%, and that obviously affects the economy. So current regulations are based on the cell count, and there's actually no test for toxicity in the water. And this is an interesting area of discussion because we don't know if the algae might be classified as toxic, however they might not produce the toxin. And here's someone taking the water sample from the algae. As you can see, it's quite dense, and this is how some cell counts are performed. So our project, our 10-week project that we sort of came up with, is ToxiMop. So one of the toxins that's produced by algae is called microcystin. Microcystin binds to protein phosphatase 1 and inactivates. And protein phosphatase 1 is something that's stuck in the liver. So what happens is it can cause liver failure. So protein phosphatase binds to microcystin and inactivates it. But actually microcystin inactivates protein phosphatase 1, so they're both kind of useless. Which we thought was very interesting, and this is actually a picture of protein phosphatase 1 and the microcystin there. So we thought to exploit that, and we engineered a bacteria that produces human protein phosphatase to bind that, and then by removal of bacteria we would also remove the toxin. And as you can see, here's the ToxiMop here. It looks like a little bit of a tea bag, and here's John holding the mop as well, like an actual mop. And this is the Moptopus. It's a hardware device, and this is sort of where the engineering, mathematics, and computing really comes together with the biology. And as you can see, we've got a lot of stuff attached to it, and there's a pH meter, we've got a temperature meter, a light meter, which will all predict the possibility of an algal bloom occurring, and it will provide data on demand as well. So this box will sit in water and actually monitor everything, which is very good because it provides data on demand. And also alerts the public via social networking sites. So if there's a possibility of algal bloom occurring, it can be like, oh, look out guys, sort of possibly post it on Twitter and on Facebook. And also another clever thing, which is sort of not mentioned here, our mop, upon binding of the toxin, will start to produce light, which will then be picked up by this device, so recognize that there's toxin in water as well. So thank you very much for listening. I hope this was a very informative presentation. I would like to thank our sponsors, and I'll now pass you over to Rachel and Chris, who will chair the debate. Thank you very much.
So Thank you guys for coming. My name's Rachel. I'm the mathematical biologist on the team.
I'm Chris, and I'm a molecular biologist on the team as well. Okay, so first of all, I'd just like to say thanks to everyone for coming along. I really appreciate that. It's been fantastic that so many of you have come along. The reason that we've invited each person is because we value the input that you hopefully bring to us. And also with a unique set of experiences from different sort of areas with different concerns probably regarding this issue. So we look forward to hearing what everyone has to say. So it's going to be a discussion relating to algal blooms as a problem. Also we'll be talking about Clatto specifically. It's a good example of a local issue where algal blooms have caused problems for a number of years. And we'll also be talking about synthetic biology generally, and more specifically our project. And this is where we most like to hear what you guys have to say, so that we can hopefully improve our project and tailor that to sort of the concerns that you might have. So some ground rules, we have some time constraints, so forgive us if we need to be rude at times and just move along to the next part. Okay?
Just so that everybody knows who everyone is, you go around and like your name, just explain what your profession is and exactly what you're here to explain today. So Philip, if you want to start.
Okay, my name is Roger, and I am a member of the Dundee iGEM team. I'm here to represent the team on the panel, and I'm a molecular biologist going into my 4th year. Okay.
Hi, I'm Andrew Landwarn. I'm the coordinator of Friends of the Earth Tayside, and I'm a consultant in sustainable development.
Hi, my name is John Pratt, Assistant Parks Operations Manager for Clatto, west of the town. My concerns are also the potential use of Clatto once we tackle the algae blooms.
Okay, my name is George Potts. I'm the Senior Countryside Ranger for Dundee City Council, and I started work at Clatto in 1983 and had 10-15 years working there. An experience of blue-green algal blooms and the impact on recreational access to the water.
I'm Kate Hahn. I'm currently working with George as a countryside ranger, but I was a research biochemist for 15 years, so I'm quite interested in the science that you're going to use to produce your toxic mob.
I'm Christine Edwards from Robert Gordon University in Aberdeen, where I work with Professor Lawton, and both of us started our life with algae here in Dundee back in the late 80s, that long ago. So we're interested in many aspects of cyanobacteria, so our research covers many areas.
I'm Craig Johnston. I'm a mathematician on the Dundee iGEM team. I spent the last few days up at Clatto, so I'm here today just to represent the views of the local residents surrounding the Clatto area.
And I'm Kieran Kelly, and I'm an early stage synthetic biologist doing research in this area. And my work is to do with trying to come up with alternative energy sources, green energy sources. And so I suppose I'll be trying to put a bit of input into synthetic biology and the community at large and see how everything fits into that.
Okay, thanks for that, guys. So we'll just get started. The first question I have is actually for Dr. Edwards. So we want to start just the basics to explain what cyanobacteria are. Also maybe talk a little bit about the blooms, and would you be able to explain why they pose a threat to human health and animal health as well?
Well, fundamentally, we wouldn't be here without cyanobacteria, because they were responsible for oxygenation of the planet some 3.5 billion years ago. And they survive in many niches. They have diverse biochemistry. In fact, I would consider them as the world's best chemists, and are now being used to exploit for biodiesel and all the rest of it. They produce high-value chemicals, which we exploit back in Aberdeen. We produce hundreds of litres of algae a month, cyanobacteria a month. But in certain circumstances, they get a bit out of control. Usually high nutrients, but of course some of them do nitrogen fix, which is another important aspect of their biochemistry. It's very green, and people often use them in Asia as fertilizer in the paddy fields. So they aren't all bad. But in certain circumstances, and nobody knows why they produce toxins, people are still, including ourselves, looking into why they do it. One of our projects at the minute is very interesting because we have something from another cyanobacteria that completely inhibits the toxin production, and that very all naturally, so it releases something into the water, the media. But, you know, that's the $6 million question. However, the fact is the range of toxins can have both chronic and acute effects, so your liver damage you alluded to, but also they are tumour promoters. The microsystems are tumour promoters. A lot of the problems in Scotland, and particularly when I was in Dundee, was from the more benthic oriented, so it doesn't look like a cyanobacterial bloom, but you can be in a nice clean water such as Lochs up near Aviemore, pristine looking water, not high in nutrients, but you've got this benthic, looks like mud on the rocks, and that was producing anatoxin A, the neurotoxin, and that killed a lot of big dogs. Some were rescued with a quick stomach pump in. At the time when I was here, I did a lot of different analyses and extractions of dog stomach contents, but they are potentially very harmful as well. And those are on the increase around Europe. So there are the microcysteines, the peptides we've been working with are the most common globally occurring compounds. The challenge with analysing for them, I mean there are many commercial kits, some of them better than others, depending on the cross-reactivity of the antibodies used. They have microcysteines, I don't know if you know it, they're like a little circle with a tail, and depending on how the antibody's been raised, depends on the cross-reactivity with the other variants. And there have been over, probably in the literature now, about over 90 potential different variants of chemical modifications or or other amino acid substitutions. So it is a challenge for analysis, and people think one of them is the most common one globally, but is that because people are looking for it and have their methods tailored for that? People are also finding them in lichens and other areas now. And I came across a paper the other day, they have a suspicion that they are in the marine environment as well. So, but they are globally the most problematic group of toxins. There have been human fatalities. That was some dialysis patients in Brazil. About 70 people died, and this is basically a filtration unit that wasn't getting the toxins out of the water, and they received high doses and they were compromised anyway, but that didn't help. And then the ones that survived, to follow up what happened afterwards, all the hospital records disappeared. So, but there have been a lot of human illnesses and a lot of animal fatalities.
Dr. Edwards, I should also say, if anyone has any questions for Dr. Edwards, feel free, that would be great actually. But just to sort of move on slightly to the sort of regulations that are in place in Scotland in terms of the testing. So that's carried out by the Scottish Environment Protection Agency, SEPA. And what they do is they count the number of cyanobacterial cells, and they also try and identify the type of cell. Often they only classify down to the level of the genera. And I just wondered if is that quite a general level? Could some species from the same genera be both toxic and some species be non-toxic?
Yeah, I mean, for example, microcystis species, you could probably do a survey and find of the ones you do 50% you test may be toxic, 50% may not be not toxic. But you've got to take into account where you sample from, time of day, you can get spatial and temporal changes in toxicity, and also if you've usually got multiple strains, they're not going to be all like, it's not like a clone that's in the water, so you may get changes in dominance, succession along the way. So you have there is no guarantees. I mean, some people monitor by looking for the microcystin production genes.
Okay.
But are the genes active at the time? So, you know, of the many methods out there, each one has advantages and disadvantages.
So I think at the moment, SEPA have sort of a test every month, but you can request further tests on demand, I believe. But when you're saying that things can change quite quickly, it seems that that might not be wholly appropriate, that the tests are done once a month. It's just a snapshot of a very dynamic
Meaningless.
process. Yeah, meaningless. And there's also, you will have seen this statement before, there's a couple of different figures, but apparently 60 to 70% of algal blooms produce toxins. So by identifying the cells being there of a certain type, it maybe does not indicate that there will be toxins produced. What would you say regarding that? Would you say that?
I would say it's probably right. You never know whether and also they may be low levels. There are also other considerations. I mean, cyanobacteria are gram-negative bacteria, so they have lipopolysaccharide in their cell wall, which is an irritant. So if you're, you know, that can be a problem, and it can also give you some respiratory, if you had them come in a shower, so if you were doing an outdoor shower, say from water that has that in, you can get respiratory problems. But that's not because of the microcystins, that's because of the cell wall of these organisms, and that can cause swimmer's itch. So that's historically what swimmer's itch if people swim with algae. So
can it be difficult then to determine, to distinguish whether something's been caused by cyanobacterial dermatoxin and something that's been caused by these lipopolysaccharides in the cell wall?
It's normally, so the symptoms are different. I mean,
Okay.
the symptoms with microcystins are far more serious, more like pneumonia and stuff like that, and affect the liver enzymes. So you can monitor if somebody's been exposed by looking at the various liver enzyme functions. That's what people have done. So there will be different symptoms, but it's still, you know, it's not an advantage to go swimming in a thick, scummy water.
You were speaking earlier, when we spoke before, about the sort of occurrence of these algal blooms, because we've noticed that even in the Dundee area, we've seen signposts up quite ubiquitously, quite widespread. It seems that of our few sort of recreational water bodies, that a lot of them are affected by algal blooms. So on a more national scale, how big a problem are algal blooms?
Well, it's quite interesting because we've had a student doing a media and a survey of the UK as part of the EU Cyanocost consortium, which is a group representing 26 countries in Europe, academics, industrialists, you know, stakeholders, water providers, this sort of thing, to evaluate the various problems of occurrence and monitoring and look how these can be improved for better future management and control and monitoring. And to sort of get an idea of what's been going on in the UK since 2005, there is definitely an increase in the number of events. She looked at the impact on whether it was recreation, drinking water and stuff like this. Unfortunately, the response to the survey was quite poor, which actually reflects the fact that people either haven't got the information or don't want to tell you. And in my long history of being this, particularly with drinking water control and drinking water inspectorate and water companies are very cagey about the information they give. And I've worked for the Water Research Centre and some other water companies, and we've also done a lot of water monitoring, which has exceeded WHO guidelines by quite a lot.
That's really interesting to know. Yeah. Is that regarding toxin levels or the
That's regarding toxin levels. I mean, from our point of view, you know, all our research is very much biochemical and analytical. I mean, to me, counting cells and looking at things, I like to actually have proper data on what's in there, chemical data, quantified and qualified. So we routinely use UPLC with mass spectrometry.
Yeah, so you wouldn't be interested in indirectly inferring something about toxin levels by counting the cell number of in your sample. You would just go straight to measuring toxin levels.
For our sort of interest, yes, but if you were a stakeholder or you know, you have the reservoir at Clatto, there are some quite good kits that you can get, immunoassays that you can do in the field. And in fact, we were involved in the production of some dipsticks, which is just like a little pregnancy thing that you can do by the waterside, as long as you've got some hot water to lyse the cells, so you get the total. Because you've got to remember that most of the time the toxins are inside the cells, unless it's cylindrospermopsin, that's a different story. And only when they lyse, break, does it get released into the water. But you need to kind of know both, or the total amount, to get an accurate amount of what's there. So there are...
Are these kits expensive? It's affordable?
Well, we don't buy them because we've got our own bits and pieces that we do, but they're not overly expensive, probably a few £100 for use. But you either get a colour change in a thing or a line, or the absence of a line, but they are all based on either 10 micrograms a litre, which is the WHO recreational guideline limit, or 1 microgram per litre, which is the WHO drinking water guideline. So they have like a recreational one and a drinking water one. So that these tools are available, and usually the best scenario is to have a quick test. If your test proves positive, then it's good to then do the more complicated analysis. But if it's negative, then you can be happy. But the word of caution is you don't have a single test for all the different toxin types. So you can get immunokits for cylindrospermopsin, saxitoxins, I think anatoxins, but I'm not sure, and microcystins. So the challenge is, it's usually microcystins, but you can't guarantee. And there are global changes in both marine and freshwater areas of the kind of dominant species. And you know, at the same time, there's also new toxins potentially coming along. There's a paper published a couple of years ago in Environmental Science and Technology where they looked at toxicity of a microcystis strain from the Pasteur culture collection and purified toxin, and actually there was something else contributing to the endocrine disruption affecting upregulation of beta-logen and lots of other genes. So something else other than the microcystin was causing a serious effect in the zebrafish they were using as their test organism. And there have been subsequently other reports of natural endocrine disruptors from cyanodes. So that's just another aspect.
Just briefly, because I'm quite interested personally, but what sort of explanations have people come to regarding why they produce the toxins? You indicated that was quite speculative.
The $6 million question.
Yeah. We heard that there was something to do with maybe competition between, there's so many different species of cyanobacteria, maybe they're in direct competition and there's an arms race and they're developing these toxins to outcompete each other.
No, well, the fact that they are inside the cell until they lyse would suggest that there's not really a competition. But it's a bit like, you know, people have made assumptions about antibiotic production in the bacteria, that that's a competition thing. But now, as people get looking a bit more closely to actual what is happening in the environment and not in an agar plate in the lab, the concentration levels that are detected and then how much aren't toxic to other bacteria, and actually there's more of a signalling role inferred. There is an indication that they might have a role in photosynthesis, protection of various enzymes and stuff like this. Some people think they're signalling compounds, but again, for signalling compounds they have to be excreted, otherwise. So it's just one of these
It's
Yeah.
very interesting debate. It really is.
And it'll be a very good paper when somebody sorts it out. We're doing stable label studies, looking at de novo production and turnover, which is it's an expensive set of experiments because it costs a few £1,000s, 100 grams to work with the nitrate, but we've got some very good results. But there's still a lot of work to be done now.
Thanks a lot for that, Dr. Edwards. I'm actually going to move on now to speak about Clatto. We've got the senior countryside ranger, George Potts, here. We've had an interview with you before, but for other people who don't know so much about Clatto, it'd be interesting if you could just explain how algal blooms have affected Clatto, how long has this been happening, and what is the state of Clatto now?
Clatto was a service reservoir used as part of the public water supply to Dundee, and that fell into disuse in about 1975. Became a country park in 1979, and was used as a recreational body of water for rowing boats, windsurfing, sailing, and attracted a lot of swimmers as well. First became aware of algae in the middle to late 80s, and Professor Codd from the university here in Dundee came and took samples, and some of the early work on the toxicity was carried out with Clatto water samples. The impact historically that it had was to close the water body to recreational use for periods of time. That tended to be, unfortunately, during the summer school holidays, so July into August. And it could be closed for 3 or 4 or 5 weeks at that time. Sometimes, depending on the type of summer that we had, it would be closed for a couple of weeks, open again and closed again. So it did make life a little bit difficult, but it didn't cause a great deal of public alarm. The water looked particularly uninviting at the time of the algal blooms, and so there wasn't any great temptation to go into it. What's happened since then, probably about 96, 97 onwards, is that the threshold that was used to close the water was brought down to a very low level. So this World Health Organization level of 20,000 cell count. And
that's per mil of the water sample, isn't it?
Yeah. And Clatto achieves that quite easily, probably by the end of May. And so over the last 5 or 6 years, as you mentioned in your introduction, then that's meant that Clatto's been closed from May, the start of the water sports season, right through to September, with no great prospect of it being reopened within that period. So that's the impact it has had. And then without being able to promote the water sports, without being able to promote the recreational resource, then that's had an impact on visitor numbers. In addition, some of the signage that's required and some of the less capable reporting that's happened in terms of media coverage have given the indication that the park is closed because the water is dangerous. And that's deterred people as well. And then, although some of it is anecdotal and some of it is factual, people believe it's dangerous to their dogs, and that's one of the popular activities.
It can be very hard for people to distinguish as well if they're not informed in the area. It can be very difficult to make sense of the reports from newspapers, for example, or more accurate information that you would work with, Dr. Edwards.
So the headlines tend to be verging on the water is dangerous, and by implication, attendance at the park is dangerous, and that's had an impact on visitor perception and then a consequence visitor numbers and visitor activity.
Just while we're speaking about visitor numbers declining, Craig, you went to speak to people who were walking their dogs around Clatto and some of the residents who live in the nearby area. What was the general sentiments of people? We did try and get some residents to come along, but no one was available. But Craig can
So the general idea was everyone we spoke to had an actual opinion on the issue of Clatto. So there was no one who wasn't interested in the water body. Everybody in the Sound area had an interest in what's going on there. Everybody we spoke to wants to see something done about it. Everyone's in the same boat in that sense. the general ideas of people were quite contrasting. We had some people who continually walked their dogs there. There was one person we spoke to, he was saying that his dog, he jumps in the water, he swims in the water, and he even drinks the water there, and he's been going there well over the last 6 years, and he's had no problems with his dog. So he was certainly of the view that these signs and warnings were quite ambiguous, and he didn't really feel there was that big a danger for him personally. So he was quite suggestive that the regulations were a bit speculative and issues, not quite correct. But there was other people who were there. There was one girl we spoke to, she claimed that her dog had went into the water and came in this rash on his belly, so she'd now...
so she's not been back for
yeah,
6
she's
years.
decided not to go back at all. She had one bad case, and she's decided not to return to the reservoir. And then there's other people who avoid going to the reservoir as well, just because of the danger of the water, the so-called danger, if you like. And there's people that do go around the reservoir, but just don't allow their dogs to go in the water. And then there's other residents who use the surrounding woods, but kind of tailor their walks to avoid going near the reservoir because of the danger that they feel is at present. So there's a lot of contrasting ideas on how safe, how dangerous the water is, and a lot of contrasting experiences. But it was interesting to see that one dog had came up in this rash, and this other dog who'd been going in over this time hadn't had any side effects. I don't know if that ties in with what you're saying about the change in levels, and then so can we justify the
It could also
regulations?
be the dog, maybe it could be an allergic response as well.
Yeah.
Some
The
breeds of dogs
lipos and the lipopolysaccharide, it might not be anything to do with the toxin. It might be just the cells themselves can actually cause the thing. And 20,000 cells is considered as low risk.
Yeah. And that threshold, that has a low risk. We've no idea of the level of toxins that 20,000 cells can do. It might not be toxic till 100,000 cells or 200,000. We just don't know, and we don't do toxin tests now. We just do this basic cell count, and even the World Health Organization described us as a low safe level. And it's related to drinking water and not related to recreational water.
Can I ask you a question on that? So at the moment, if you detect algal bloom, is it the remedial course, so the way of clearing it up, is that just a case of leaving it to basically sort itself out, and I guess wait until the weather gets a bit cooler outside, and therefore you can do the cell counts and they drop below that threshold? Is that the current kind of approach to fix things?
Yeah, we don't have a fix in place at the present moment in time, unfortunately, but yeah, we'll always wait until the next set of tests go in. We really just react on the count that comes back, and obviously the advice we get from CEPA, whether we close the reservoir or take the notices down, whatever.
Yeah, interesting then. I think that leads us on to, they've tried a couple of things at Clatto, haven't they?
Yes.
Maybe you could explain those, and maybe Dr. Edwards can give us more information on those as well, as mechanisms for dealing with the issue.
Okay, for several years we tried using algal control with barley straw. And so we made straw sausages. They were contained within mesh tubes that are used to package Christmas trees. But they were long sausages with floats in them. Gave a big surface area of barley straw, and apparently the degeneration of the barley straw releases some kind of enzyme which inhibits blue-green algae growth. Okay. That was tried, but that was fairly labour intensive, and it had limited effect. It did seem to suppress the algae, and it's a method that's been used elsewhere. The next thing that was tried was a business called Phoslock, which was adding clay particles to the water, which then bound to any free phosphates that were in the water and locked them up, so that they weren't available as a nutrient for algal growth. And that had a really remarkable effect on the water. For several months, right through the summer months, the water stayed very clear, visibility 2-3 metres. The great thing about that is that the aquatic plants were getting light, and the weed growth was quite substantial, so that your LDS and your other common pondweeds, their growth was quite strong. Unfortunately, people equate that with algae, and so it was full of weeds which were dangerous, and it wasn't safe to go near the water because of these pondweeds might grab you.
Yeah, well, they're the same colour, so.
Same colour. That year, the first year after the phosphate treatment, there was a late algal bloom in September, which was unusual, but it was also unusual in that many other water bodies, from Rutland down in England to Roscommon as a local water, they also experienced a late algal bloom, an unusually late algal bloom that year. So climatic conditions do have an influence on that.
Maybe Clatto also reflects what you were saying, Dr. Edwards, about this trend towards increasing the occurrence of algae blooms across maybe Europe. Is that across Europe?
Globally.
Globally, yeah. And Clatto reflects that almost. It's becoming more of an issue, and maybe it's getting warmer as well.
Well, I'm just wondering because in the early years, 83, 84, we had fairly high water temperatures into the 20s, into the low 20s. I just wondered, I was going to ask Dr. Edwards about the influence of water temperature on levels of toxicity. And but the year after the phosphate treatment, it was just a normal summer. It wasn't particularly high water temperatures. You have to bear in mind that we're fairly northerly, 56 degrees north. We've got a short summer season, and water temperatures above 20 centigrade are not prolonged and they're not common. So in terms of algal growth, I don't think we have the conditions to produce particularly dangerous blooms.
They have lots, and I've worked with people in Sweden and Finland and Norway, you know, even further north. There's, I think everywhere there are algal problems, and partly it just depends if people are working on them, but definitely further north than we are. And even in the Antarctic, there are toxic microbial mats.
It's interesting, yeah. George was starting to speak a little bit about some of the things they've tried. They've tried a few things now, with some levels of success, usually temporary. What do you think about those techniques? Are there other things that George could try at Clatto? What's available to people who, you know, there's a lot of water bodies that are closed for recreational activities. Is there any options open to people to deal with the issue?
Well, there's physical ways of removing the cells, like using booms. People sometimes remove scum manually. I've got pictures of that. Copper sulfate causes lysis. People have used that historically, and there are other algicides that are probably a bit more
that's probably should actually say,
friendly.
yeah, to bring Andrew in from Friends of the Earth. You would, I guess, you'd want to limit the impact you have on the environment that you're dealing with as well. If you're going to be, these cyanobacteria must form some component of the normal ecosystem.
Well, they're absolutely
by eliminating
essential.
them might not be a
They
good
are
idea.
essential in the food chain.
Yeah. So
You don't want to eliminate them. You just want, I mean, ideally, good management strategies and then watershed management. And I know from Roscommon, for example, that farmers in the area have been incentivized to use environmentally friendly fertilizers, but they don't.
are they more expensive?
Yes.
Yeah.
But they have been incentivized, so you know, they get some.
Discount of some kind.
So I don't know
Support.
quite how it works, but I know from going to it, because we use Roscommon as a sort of material for some of our experiments. One thing I would say is that upon lysis, if you have microcystins, for example, in particular, which we've worked with most, they do generally rapidly degrade. I mean, you've got a rapid dilution factor anyway, but also they do rapidly degrade by microbial action, because we're just in the process of.
We've seen some of the work on that as well. The half-life of microcystin can vary quite a bit depending on
Which one and which content, how much and stuff. But we're actually, we've discovered a universal and even from, we've got strains of bacteria from places that have never been exposed to microcystins, rivers, but we've got some good degrading strains from there. But they seem to have a universal peptide degrading mechanism along the lines of typical bacterial antibiotic resistance. So you can throw in all sorts of different peptides, whether it's microcystins or some of the other ones produced by cyanobacteria, or antibiotics from Sigma, or even, you know, things, human peptides and stuff like that. So that which opens up some possibilities for simple water treatment options. And there are quite a lot of international groups looking at biodegradation by augmenting sand filters, so simple treatment solutions that can be effective. So those kind of things.
Thanks a lot, Dr. Harris. I'm sure George will have a few questions regarding potential things that may work. We've tried quite a few things, haven't we?
Well, yeah, I was just coming to mind that there was other tactics that we've tried and I've been aware of. One is lowering the water level to allow the frost into the margins, was the idea that would disrupt the spores that were in the mud. If you've got exposure to low temperatures, -5 and beyond, then that seemed to have an inhibiting effect on your algal bloom the next year. The other one that's quite commonly used is a throughput of water. So Clatto tends to be only rain-fed, and if you run a supply of water through it, then that helps to reduce the amount of free nutrients that are in the water, and that can reduce your, inhibit your algal bloom as well. And that's quite commonly used, a throughput of water.
Yeah.
And there are other tactics that I've seen is turning over the water column. That's used at Lintrathen by oxygenating pumps, and so they're based on the floor of the reservoir, and they turn over the water column by circulating the water.
That's quite common.
Can these things be quite effective as well?
They can be, because usually the blooms occur in a stratified water body, and if you disrupt it, they don't sort of respond the same way, and they don't end up, because when they're particularly microcystis, it's very gelatinous, and they're single cells, but they're in like multiple colonies with a mucilage around it. It's like a mobile biofilm, if you like, those such things. But there are other strains that are quite common that you never see on the surface. Clatto likes to be lower down, maybe a few meters down, and people often find that's nicely situated to the water intake for the abstraction point for drinking water treatment. They seem to be more common in France than anywhere else, but it might be just because the people working on it are in the Paris area, and that's what they've got. But they are now finding that they've got these Planktothrix blooms at 5 meters down all year round, whereas again it used to be seasonal. So
interesting. A lot of unknowns.
A lot of
this area,
unknowns.
yeah. Just to move on now to speak about the project. So we've got Philip here from our iGEM team. So I would ask Philip, can you explain why we've chosen the project briefly, but just also explain what our project is, and maybe a little bit about iGEM and what it is.
Okay, we are part of the genetically engineered machine competition. It uses synthetic biology. So it's like, it takes a lot of the techniques of genetic manipulation that have been around for a while and tries to standardize those, and it's mostly used in bacteria as well. So it's useful to other people, and they can come in and make composite parts out of things you've made because you've done them to a certain standard. For our project, we chose to focus on these algal blooms because there's a clear local issue. A lot of the water bodies have been shut for quite a while during the summer because of them and the toxins which are released. So we thought there could be a place for a synthetic biology approach to this problem, to remove the toxins cheaply and effectively, and hopefully to find some way of detecting the microsystems specifically. The reason we think that the synthetic biology approach will work is because it uses bacteria, and bacteria can be replicated very cheaply and in large numbers. So we want to produce a proof of concept that this sort of approach would work. Specifically, we chose microcystins because it binds PP1, and the interaction's been studied quite well. And it's also a very common toxin produced, and it's also very convenient because PP1 is also studied within the university, and we could easily get the gene sequence for the protein which binds the microcystin.
So what exactly would be the approach then with this project? So
okay.
How are they going to utilise that interaction for the project?
Okay. The bacteria that we use have a membrane that's in between the cell as a whole and the outside. So we're planning on putting this protein which binds the microcystin within that membrane, so as to mop up microcystin, since the microcystin can get into that membrane. It can bind there, and it will so be sequestered by the proteins in the membrane, and then we can have the cells in some sort of containment with water contaminated with microcystin flowing through it, and so we can keep the cells within a confined space, and also allow us to mop up the microcystin, and we could then remove the cells along with the microcystin bound to the PP1.
So quite
that would be
simply
the
then, you get these bacterial cells, E. coli cells, to express PP1. Part of the challenge has been trying to get the PP1 protein to go to the periplasm, the space between the two membranes on the outside of the cell, so that hopefully there'll be more interaction between the toxin and PP1. But yeah, Dr. Edwards.
But you're making the assumption that you've got soluble microcystin.
Yeah.
So you,
yeah, it would only bind soluble microcystin. It wouldn't bind any that are within cells or not free.
but most of the time the problem is in the cells. And if you can do something, I mean, ultimately if you can remove the cells safely, then that's before they lyse, and that's a more ideal situation. I mean, it's still an exciting thing, but it's quite challenging. How far have you got with it?
We've been able to put the PP1 into the periplasm, and we've run tests very recently using microcystin, and there has been some effect. So we've been able to use what's called an ELISA to tell how much microcystins are in there in the water samples. So we have been able to sequester some of the microcystin within the periplasm with the PP1s there. Currently, it's we want to focus on trying to improving the amount of PP1s getting into the periplasm, so as to increase the mopping ability of our cells.
And what about the fact that are you planning to test other? Have you just used microcystin LR?
Yes, that's right.
And are you planning to evaluate the other ones, or a set of other, because they have quite diverse solubility characteristics, even from a one amino acid substitution.
We would like to have studied that, but the kits cost quite a lot of money, so we only had enough to maybe test maybe one, and we've got quite a lot of things.
Well, maybe we could have a chat.
Okay.
The other thing is that there are other cyanobacterial peptides that inhibit protein phosphatases. Have you considered this?
Yeah, we have thought about that, but we haven't been able to focus on testing whether or not they have.
So even
Whether or not
buy,
they
for
can
us, even buying in
detect
the
it.
microsystem was quite expensive. So that I think is it the nodularins toxin as well, we
Mhm.
bind to PP1, I think. And
Yeah. And
so there's a few other
aminobutyl
ones.
peptides.
Yeah.
From a
Collaboration, you see.
Yeah.
You have to develop, this is a useful thing for you, is developing scientists is be cheeky, nicely cheeky. And when I was here at Dundee a few years ago in the postdoc, we didn't have nice toys at the time. I mean, we weren't too badly off, we had HPLCs, but I've always wanted a mass, like you get mass specs on the end of them is so much nicer. And I just contacted somebody at GSK who'd published a lot of LCMS work on the marine toxins, and I just said, hello, we've got, we've seen your wonderful papers in this, that and the other, and we were really wondering, we've got this really good separation method, but we had some bloom samples from X, Y and Z, and we got ran the samples, had several publications, that's obviously you're young and learning, but
I did contact Professor Lawton to try and get some microsystem, but I didn't sadly get a well, I did get a reply in the end actually, but I think it's been quite a busy time for her.
Oh yeah, we've been moving buildings, it's
moving
been
buildings,
a bit bonkers.
so we weren't able to get any. But we have got a lab that works on it within Dundee University, and they've been really helpful actually. So I think the thing to probably clarify regarding iGEM is it's this synthetic biology competition. It's all about trying ideas. It'd be unlikely that this thing is going to be mass-produced or manufactured and commercialized or anything like that. But what can be useful is this principle of maybe having a protein that sits on an outer membrane due to its strong interaction with something that could be a toxin, or then this could be utilized to make some sort of mock device with bacterial cells. So it's just about sort of proving that that could be usefully applied, and hopefully other teams in the future could try and apply it to other problems.
And that's the interesting kind of thing to add on to that, is that in synthetic biology, one of the biggest kind of changes of change of approaches has been this idea of actually having a public repository for parts. So the idea is that you take a useful biological system or something that's found useful, for example, like you mentioned with the species of bacteria that can naturally degrade these microsystems. For example, that might be a really difficult bacteria to work with, to try and manipulate or do anything kind of from an applied point of view. And the idea is to take obviously the parts that are useful, the parts that might actually affect the microsystem, when they become well studied and well characterized, and try and use them in maybe in a simpler system.
Yeah, we put the Rhodococcus gene into E. coli.
Yeah. And then you can try and increase the activity, or you can take it from something that might be slightly it might be a bit dangerous for the environment or in itself, so actually making the problem worse. And in that sense, iGEM has been great, because iGEM and everybody who takes part in iGEM and builds these systems, they actually have to give these parts to a public repository, which is actually available then to other people in the field who want to use that part in the future. You give up automatically your claims on patents for it. However, if you take a part and combine it with something else, and you actually manage to construct a useful biotechnology kind of tool, you can then patent that, and so it's still driving kind of commercialization of these things. And actually, that is one of the biggest drivers of obviously a lot of innovation, is actually if you can make some money from it, and actually make a useful product that actually gets out there in the end. And I think that's really interesting, the fact that in a 10-week project you maybe contribute these parts, and then someone can then develop on further on that in years to come.
There's a lot of mileage. And actually, I'm sure that approach is so practical from stopping people reinventing the wheel and going, yeah. Because I'm sure a lot of that goes on. Well, I know he does.
Yeah, absolutely, yeah, no, it does.
Andrew, so we're starting to speak about iGEM, so it's the synthetic engineering of biological systems, to try and use tools to deal with problems that we hope that, obviously, as a member of Friends of the Earth, that you'll be interested in. So we have a clear common ground between all of us that we're interested probably in environmental issues. Is a synthetic biology approach something that you would see as ethical or reasonable to deal with a certain problem? Is it something you would feel comfortable with?
Well, I think we've got to learn from previous efforts to introduce new innovations in kind of dealing with life systems, and the fact that sometimes they've not been fully tested, and often, I mean, as with most environmental problems, they result from the unintended consequences of well-meaning actions. So we decide we want to do something to improve some aspect of quality of the environment, quality of life, food supply,
water supply.
Water supply. You've seen what's happened in Uzbekistan with the Aral Sea drying up. That was because they redirected the rivers to produce cotton. And the Aral Sea is now about fifth of the size it was. So because it wasn't getting the water, it's now just toxic salt pan, most of it. And it used to produce a lot of fish for the people who lived around it. And now they're getting infected by the
you would be quite interested in to try and preempt potential consequences of any application,
I think,
try and work out what could happen if something goes wrong.
Well, I think with anything in life, where you're doing something and you're not quite sure, you've not done it before, you really ought to do a risk assessment. Risk assessment is one of the most basic things in any organisation, in any innovation in life. You do it yourself when you're going to put a pan on a stove. You think, well, what happens if that boils over? It's better turn down the gas, you know. It's a basic thing. It's thinking, well, what could go wrong with this? What haven't we thought about? What could happen with this if it gets released into the environment? So how can we be sure it's not going to get into the environment? Or we may say it's benign, it gets into the environment, it'll die within 12 hours and not affect anything. Well, you've assessed it, you find out there's not a risk. But you have to go through that process, and with a lot of these innovations, that process has been ignored, and there's a tendency to focus in on the problem without thinking about the systemic context of that problem. So what has given rise to that problem in the first place? You've been talking about the fact that there's excessive nutrients in the water. Well, why are there excessive nutrients in the water? You talked about other types of fertiliser that might affect that. The problem with fertilising systems or approaches is, with a lot of other things, the costs to society have been externalised. If the farmer buys the fertiliser and sticks it on the land, he's not paying, he or she isn't paying for the cost of that on Clatto Reservoir and the cost of having to clean it up.
And of course,
so
if you want to use...
If you did pay for that, then the cost would go up and the market would adjust, but the market doesn't work. It's a problem with economics. You need an economist on your team as well to think about, well, what's the economic system within which this operates. And it's really thinking about this as part of a system. You've got a problem here which is a result of a malfunctioning environmental ecosystems are not working properly because we've screwed them up, because we're putting stuff into them which messes them up. So do we deal with the problem that's resulted from that, or do we go back and try and deal with the original cause of that problem? Because it seems to me if you deal with the toxin, whether this, there's one of the questions you raised, the first question you raised is there a problem with with what you're introducing into the environment to try and treat the problem. It's an organism which you've played around with. You've got to assess that and whether that's a danger, a risk, what that could do. But you've also thinking, well, is this actually going to deal with the problem? Because if you've still got all this messy algae in there, is anyone going to want to use the water anyway? You know, even without the toxin, they're not going to want to go in a kayak in Clatto and risk getting their hand or splashed with this muck. So it's not going to solve. It's going to solve one problem, that the dogs aren't going to go in and get rashes maybe if the, but you've still got the problem. So is there an opportunity there? Can you mop it up literally, capture it and turn it into biofuel? Or is it too expensive to harvest? Or think about the wider context of this, and maybe what you're talking about will deal with one of the consequences of the systemic malfunctions that it helped to detoxify the stuff. But you're not going to get people using it without getting rid of the stuff in the first place.
Yes.
And maybe that stuff has a value. You know, so many waste products in society, which is an issue in Dundee at the moment with the recycling centres. You know, a lot of waste products which can be resources, useful resources, if we can find a way of using them. And maybe this problem we've got, we could turn around into a benefit.
Yes, it's a clear appeal for that approach. So I'll let you come in, Dr.
That's a good point, and actually you can remove them, and people who are using algae for either biodiesel projects or pigments, I mean, they are finding a lot of biotechnological application these days. We're interested in the high value compounds area, but they are wonderful for other things. We're also looking at biodesalination using cyanobacteria. But you can quite simply, if you use standard alums that you use in water treatment, you can flocculate them to remove them. So that is something that can be done. Or you can use some filtration systems.
Kieran, just to bring yourself in, so Andrew's talked about risk assessment. Could it be that by highlighting or emphasising the complexity of living systems, you stall any development beyond the risk assessment?
I think the lessons learned from kind of related fields like nanotechnology and GM, say crops or GM organisms, I think using lessons that were learned in the kind of, I suppose, in the failures to get those industries off the ground, especially in Europe. I think a lot of people are very wary about charging into developing these new technologies and manipulating genes around, and synthetic biology in a lot of ways was actually is a rebranding of a lot of these things, but actually with a real kind of substantial emphasis on security and engagement with the public, and actually finding out what people really want. For example, there is a problem, and trying to actually address a problem instead of trying to create a problem, if you know what I mean, like to make money out of it. I would say, for example, about 10% of the community involved in synthetic biology are actually social scientists, psychologists. There are some economists in there as well, who are basically very involved in kind of in making sure that the field is actually truly engaging with the public, and not just spouting things out and saying this is correct and trying to force something down their throats. So I do think you do need to do the risk assessments correctly, and I think that actually has to be built into every single project. There is an awful lot of emphasis on that. For example, if you construct a synthetic system, one of the key questions, the first questions you will get asked is what happens if this does get out in the environment, and I think that's really important. A lot of people have tried to come up with different approaches, for example, things called kill switches, so that it would lyse the cell, for example. That has its own problems in itself because you can reach genetic material. Another alternative might be actually just to degrade the genetic material straight away, so you can't have this horizontal transfer of genes, which is probably maybe a more useful situation. Containment, so if you could actually really have a, like for example with, I know you guys have the MOP system, how are you going to contain that to make sure that the bacteria that you're using aren't going to get out into the environment? That's really important.
So I'll just get Phil to come in there then. What sort of containment, what approach have you tried to use to avoid releasing a genetically modified organism into the environment, and is it effective?
We've tried using dialysis bags, which are bags with lots of little tiny holes. The holes are too small for bacteria to go out, or of the proteins, should any of the bacteria lyse. But small enough so that water can flow in between, and also the microsystem, since it's very small. It can
so inside the bag you would have the cells, and they would be expressing the protein that would bind to the toxin.
Yes, that's right. And then hopefully you could just lift out the cells and dispose of them safely. So there shouldn't be any environmental problems with that.
So Andrew, you made some great points about risk assessment earlier on, but do Friends of the Earth, they wouldn't accept a genetic engineering approach to any problem, really, would they? I've just had a look at the website. It doesn't seem as though they would. You don't necessarily need to have the exact same views.
I think the problem with genetic engineering is it's tended not to consider these wider effects. It's been driven, well, what we've seen and what's been blocked by decisions of governments in Europe particularly has been the commercial application of GM for crop production, which, or for, not for crop production, but to suppress weeds and to suppress insects, whatever, but to boost crop production without apparently really thinking about the consequences, because the environment will respond to a change to some element in the environment, so it could have all sorts of knock-on effects, and it doesn't appear to be, doesn't seem to have been sufficient analysis of these in some cases, and the problem is that every attempt to introduce these kind of techniques tends to be tarred with the brush of the company that comes in and tries to sell its products, come what may. So
again, because of the scepticism towards genetically modified organisms, it's allowed companies like Monsanto to have a huge stranglehold of the area. The companies become larger. There's probably not the competition from European companies that there's Syngenta in Switzerland, but that probably doesn't really help the situation, because companies like that probably have more freedom to do whatever they like, it seems.
Well, they do in other countries. They're selling these seeds in India, and then the people farmers in India are trapped to use these seeds, because if they're not allowed to take the next year's seed and sow it themselves. They've got to buy the next lot from the company. So there are all sorts of ethical issues which are not just about the genetically modified organism. It's about how it's impoverishing farmers rather than helping them. So there's an awful lot of issues.
Genetic engineering doesn't intrinsically cause any disadvantage towards them. It could be something that's used to help the people who need food the most. I mean, it's all about utilising it. It would seem to work in that way rather than.
Well, at the moment it's totally commercially driven with no apparent ethical consideration, as far as you can tell by companies such as you mentioned. But I don't see why the idea of genetically modified organisms properly controlled, where all the risks are properly analysed and understood, used for benevolent purposes to clean up toxins in water supplies, I don't think there's a kind of sense of, well, it should never happen, provided it's managed. I mean, there's all sorts of things being done which, if they're properly controlled and managed and assessed before they're introduced, then they can be introduced safely, or the risks are minimised. So you then weigh up the costs and the benefits, or the risks and the benefits.
And that's the important thing, to weigh up the risk and the benefits, and sort of detail the risk as best as possible, to make an informed decision. I
had a kind of related question, which was actually going back to say the Clatto Reservoir then. So do you think, with your opinion then, do you think the problem of the closure of the water body is sufficient to demand this kind, like that kind of approach, and would a synthetic biology approach, for example, or something related to that, would that be acceptable, do you think, to people who go and visit that, if it means that the water body was open again? That would be kind of an interesting question.
If that was part of a number of approaches, and so to stop nutrient loading, surface water runoff coming into the reservoir, to look at clearing up the algal scum when it occurs, and then to mop up toxins as a joint approach. I think each of these, I think it would be more acceptable if we were tackling all the angles. But just to only tackle the toxins, it's fine in the lab at the level that you're talking about, but to move on to huge volumes of water, you're striking a big difficulty.
I was going to ask if the group had calculated how much protein phosphatase they would need to do, how much microcystin per.
I think you're
and how you were going to apply it, Has that been done?
That's something that we're working on at the moment, and we, as Phil spoke about earlier, we're looking at ways to increase the number of PP1 in the periplasm to then reduce the number of cells which are theoretically required to then mop up certain levels of it.
But are you going to like drag it round behind the boat, or how are you going?
Yeah.
You know, how you're not going to throw it in. So you know you've got an area. How do you know where to go, and you're obviously going to use your ELISA to guide you as whether there's stuff there or not.
This is one of the reasons we've held this discussion today, to try and get ideas together. So it's something we did consider initially, but we've not focused too much on this sort of idea in recent times. We've been more focusing on kind of the lab stuff, making sure that the microsystem is binding to the PP1. We've not actually considered.
The PP1 costs 200. Last time I bought it, because I do use it in assays. If you buy it from Sigma, it costs 250 or 300 quid for 25 micrograms.
Yeah.
So you might be better off just selling.
That's a really valid. It's a really valid
You know,
point, and it's a great.
And that can actually, well, you could do that. You could sell it and also use the money to fund a PhD project for somebody.
I mean, developing a cell-free system. So you could try. I mean, for example, taking it and mass producing it through in a controlled environment, for example, in a lab, in a large fermenter scale, scaling up to industrial levels, trying to bring the cost of that down itself has a lot of merit actually, and then you could try and actually use that in your MOP system, which is another alternative to mobilize that protein to something, to a mesh or whatever it would be. Again, so you're taking away the idea of releasing an organism into the environment and actually having a cell-free system.
Which people are using with some enzymes in water treatment, looking at novel water treatment approaches.
They use it for arsenic, for example, and these kind of things. And so
that engineering and that technology exists somewhere.
It does, yeah.
And so it'd be a case of taking your system and seeing how it can be adapted into the existing. Because there's an awful lot of work done on water treatment across the world. Drinking water is a key component of life, and so there's a lot of time and energy spent on getting the system to work effectively, and bolting on your system onto that, finding a way of adapting that would be a useful way forward.
Can I just ask, when last time Clatto used any sort of approach to try and deal with the system, or have they kind of sealed it as redundant? Have they been trying anything in recent times? Or?
The first lock was the last one, the last treatment, and it's unlikely to be repeated because of the cost of it and the limited effect that it had.
So how many years ago did that take place?
6 years.
Yeah, about 6 years. Time goes fast.
Yeah. No, I was just interested in trying to understand why they've not been trying any new ideas with the current solution.
It's a cost. So this was a German company, and we were looking to get into the UK market and have successful pilot projects that we could showcase.
Yeah.
So the other one is Flemington Loch, Aberdeen way as well. So I think as an angler, I'm aware of Loch Leven, and that suffered terribly from algal blooms, and they addressed the nutrient loading, and they looked at ways of managing that water body, and the water quality there now is absolutely fantastic.
Yeah, it is
And that's a natural process just by cutting down on human interference and human influence. There are options there. I think where you are, you're looking at assisting recreational use because that threshold for drinking water is that much higher. And also if you go off into that aspect of it, the commercialisation kicks in, and somebody will grab your idea, take it off into another lab and spend a lot of money and not be their idea. So that is certainly a danger. Kate and I share concerns. I think the example that you gave of the seeds, I could see this going in a similar direction. If you hit on a strand of very useful science, which I think, with limited knowledge I think you are, then you could be subject to very substantial commercial pressures. That wouldn't be a good thing when you come to drinking water, and in the worst case, and lesser effect the impact on recreational access and the economic drivers that has in terms of tourism and development around water bodies. So that's a wee concern that we can.
Thanks for that, Joe. So, Dr. Edwards, since you're our cyanobacteria expert, and that's the problem we're dealing with, do you feel that the project can be useful? Maybe I wonder actually, what do you think of synthetic biology? Just as a research scientist, is it an area that comes into your working life? Or?
It's sort of on the periphery from a teaching point of view. We might be looking into it for a proposal for something else in the near future.
They're good.
Sustainable future fuel approach, the biofuels. I mean, basically, the cyanobacteria, as I mentioned before, are just fabulously full of genes for wonderful things, and they can produce, hydrogen, the nitrogen fixing, the capacity can be manipulated, and they don't need much to do it all. That's the amazing thing. I mean, even we did some work with a nodularia species, and even without nitrogen in the media, nitrate in the media, so it's fixing to provide, it's still made all this vast bunch of peptides, not only the toxins, nodularians, but also a whole range of other peptides that it produces as well, spumigins and nodular peptins. So
there's a huge opportunity for synthetic biology approaches, probably.
Oh, I'm sure there absolutely is. There's no doubt. And often what you do when you first start, but mainly your sort of plans in one direction may lead in another. I mean, even we find that, and even by doing something and making a mistake, like some of our cultures got mixed up, and then we found something that just switches off microcystin and the cyanopeptylin production. So by getting our cultures mixed up, I think that's partly because there's too many people in the culture room. We actually, but penicillin happened that way. People do, and by exploring and trying new things, you have your aims and objectives, but it doesn't, and that's fine and good. But you may end up along the way going, wow, I can sell all this protein phosphatase. But that's how science is. If it was all black and white, it would be quite boring.
Can I ask you about your E. coli strains that you've got then? Are you using strains that were already produced in the university and they just happen to be produced
Yeah,
in PD1 and you went,
DH.
that will do?
Yeah.
So it's producing soluble PD1, the whole molecule, and it's secreting it into the protoplasm?
Yeah, that's right.
So presumably the microcystin is then,
Yeah.
the outer membrane is permeable to the
You've not,
microcystin.
just I think you've missed the very start of that. You've engineered the lab strain to, I know.
Yeah, so it's a larger undertaking than maybe it first seems. It's taken us a while to do it, but to get the PP1 to the periplasm, you need to have a signal sequence on so that it will be sent to the right machinery.
But it's soluble in the protoplasm, it's not attached to a membrane-bound protein?
It should be soluble in the periplasm, that's the aim. We've tested, in comparison to control cells, the cells where it was as we've intended, hopefully in the periplasm. These cells have mopped up the toxin when the cells have been mixed with the microcystin directly in an epindome, whereas the other ones haven't. The other ones being cells that have PP1 in the cytoplasm. So it does seem, it seems to have a dramatic effect when we look at the data that this does help. And it's an E. coli strain MG1655. I'm not sure why we're using that one. But.
So what kind?
And so we had that PP1 on a plasmid piece of DNA, and so we've engineered that PP1 to have tags on it, so we can also try and locate it within the cells.
Okay, so you specifically produced the E. coli expression. Okay, I thought maybe it was already.
Just back, the MG1655, the reason you use that strain is because it's an attenuated strain of E. coli that's been used in labs for 30, 40 years longer, much longer. really well characterized, and it's the one that has the genome sequence as well. So the genes actually that are encoded by it are really well known, and it's probably the best studied of all E. coli things, and that's why I think most people start.
But that would be the standard one that you
That would be the very standard
whatever
one.
you're trying to produce.
Yes, absolutely.
So have you thought about, I mean, these enzymes that are produced by the other bacteria that are the generic peptidases that are associated with the antibiotic resistance, if you could combine your PP1 binding with that kind of enzymatic activity, then you could have turnover, actual organic living turnover. So rather than having a mop that's going to fill up, oh right, now we're going to have to throw it away, what you actually want is something that binds it, breaks it down, and can continue to function.
So that's definitely something that, through the research, we were aware of, and it's something that if we had more time, then we would definitely do that. These enzymes seem very interesting.
How long have you got left?
So it's a 10-week
Yeah, I thought you said
project.
10 week. Okay.
But this is something that the team next year might actually, in recent iGEM projects, I know for example the Calgary team, they developed something 1 year, and the team next year then took their project on and developed it further. So it's something which the team, some of us might be involved again next year, some of us might not, but it's something that the team next year can take on, or if anyone else is interested in further research, they can pull these ideas and work on it. But that's certainly something that we would definitely consider doing
That's
to
the good thing
expand.
about iGEM as well, because it doesn't necessarily mean that Dundee's team next year is working on this project, but because we've submitted the parts to the iGEM registry, another team from another university could be then working on it. They are
Showing
there,
everything.
yes.
Or you could think this is really what I'm into, and that I could do a PhD proposal for.
Often happens with the iGEM projects. But yeah, like what you guys are saying, that I think we're in an early stage where we're able now to get cells to capture a toxin of one type. And there's loads of opportunity, loads of different avenues you can take for the next steps. And I think the one you mentioned is one that has appealed to us before as well. I think it would be a really neat project to be able to do that as well. We need some more time, but it would be a really lovely project.
Yeah.
Sounds like you've done a lot in 10 weeks, 9 weeks.
Yeah, so we also have the electronic device that is hopefully a pH meter light sensor. Will work.
Yeah, how's that going to detect the toxin?
So we also have a biological device hopefully can detect the toxin, but it's very much we don't know how well it's going to work, but I feel like you could maybe explain that.
So we've engineered a receptor, so it would have, instead of the usual part which sticks into the periplasm, instead it has our BP1. So we're hoping that there will be a conformational change on the binding of microcystin, which would transfer a signal inside of the cell, and then hopefully produce some sort of fluorescence when microcystin is bound, and so would it be able to identify if there's microcystin in the
so the electronic device would recognise the fluorescence.
fluorescence. Yeah.
But it would also have these additional sensors on board which could predict, can detect ideal conditions for algal blooms.
Hopefully in that way it would be relating real-time environmental information to some sort of information regarding the bloom or the toxins that
So
are
it
produced.
was.
Again, it's a very, it's
Yeah.
just the beginnings of
It was
it.
very much motivated by this testing, which we thought was a sort of an appropriate evidence this 1 month. It was the idea behind the octopus being able to sense was motivated by the fact that we felt that the testing procedures were inappropriate. So it was motivated behind that, and it was to raise awareness on that issue as well. And that's why we've been working with George, because we do feel that the testing may be slightly inappropriate. So
What sort of limits of detection are you envisaging with that?
So we have 2 people working on the electronic device, and neither of them are here just now. Sorry, yeah, Kyle's working on it.
So yeah, it's like in the very early stages of prototyping. At the minute we're using very cheap hardware so we can build loads of them. But it's kind of limited to the size, our budget, and how much time we have. So we can probably use this one device on a tank of water and measure everything within it. which is good because we can decide how often we want to measure. So we could get data back every second and build up a huge database and then analyze that database. And if you find that, like using different like information, so you can pull like data from the web about like weather statistics, and then you can measure what's actually going on inside the lake and make a comparison. So if you find that at the start of say like May in Clatto, if there's algae starting to produce more and you've done a test to see like a cell count is like starting to raise, you can input that data, use what's in the lake, information you know, and it'll kind of do all the hard work for you and output this information that you can like kind of visualize and see what's going on. So it mightn't be 100% accurate because it would take a lot of time to develop something like that, but it would give you a clear indication of what's going on inside. And one of the things we were working on was kind of a light array, so we can visualize how the algae moves through the water. So during night and day times it moves up and down. So we'd have an array of light sensors in the water going down vertically just from the top of the water surface to the bottom. And it's kind of a combination of software development and mathematical modeling with what we know about algae. So we can kind of detect how the algae moves up and down, so we can kind of visualize where it is at the current time, and if there's like, you know, less surface light, then we can maybe detect that there's like an algal scum over the top of the water and predict how that moves. So if it becomes larger and there's less surface light, then you can kind of see that like what's going on underneath. And then using different sensors like pH, dissolved oxygen stuff, you can see if there's like the conditions within the water, and just compare everything and feed it back to people.
It could be a really useful model just as in general. You know, if you guys get a really nice model together, then in the future someone might be able to apply that to actually management or monitor.
But the key thing was these daily updates as opposed to the monthly testing.
Which is good.
And that binding interaction between PP1 and the microsystem, as I understand it, it's actually completely irreversible. It's a covalent bond.
Nodulari isn't, but the microcystin is. However, there are modified, microcystins can easily get modified chemically, and they will not bind the same way. So a methylated microcystin, for example, is 1,000 times less toxic and binds protein phosphatase less equivalently. And also amongst the different microcystin variants, they have a different IC50 with PP1 and PP2A. So that is also something to bear in mind. The variants can be quite different, and the chemically modified ones can be quite different.
I'm just thinking from the point of view of your kind of being able to turn over your mop, rather than it filling up and being able to regenerate it. If it was feasible to alter the binding site on PP1 somehow, so that covalent bond cannot happen, so you've still got enough affinity for you to actually bind it, but you're not getting the irreversible bond forming.
Like nodularin or oxidaic acid or something like, because oxidaic acid is a common tool for looking at protein phosphatase regulation. So, and that doesn't covalently bind either.
So there, as I understand it, there's other interactions between the microcystin and PP1, but the covalent bonds formed between a specific cysteine residue. So maybe if you
exactly.
take that
so
residue,
it's a complex interaction, and the affinity will probably be reduced by taking that cysteine away, but maybe that would create a situation where you can take your mop out, wash it, it'll come off, and you can stick it.
It's a really good idea. It's a really good idea, yeah.
But it wouldn't take much. It would take more than 10 weeks.
It's a really good idea.
Take those things. But like you say, the expertise is here in the university. The phosphatase work is massive. They've probably
this is
got quite a lot of
phosphatase
constructs.
heavily.
You probably have,
The other alternative is you could, if PP1 was to bind the microcystin, you could perhaps decorate the microcystin to be, or the PP1 for example, to be degraded, and therefore regenerate more of it, and degrade the whole thing together as one thing. Or alternatively fuse, for example, the PP1 to something like that does the degradation, and so it attacks it from the other side. That could be another
very,
approach.
very cool. Well, synthetic machine for degrading the toxin, holding it and degrading it would be a really cool thing. Okay, so I think we're out of time actually, so we've gone over a bit, so we should probably stop. But thanks a lot for coming. It's clear to everyone here that the input's been really valuable, fantastic actually, and we really appreciate that from
Not
everyone.
only valuable to us, but it's been advantageous to you as well.
No, no, it's always interesting.
Able to get other people's.
And who knows, they might eventually come up with something that will cure perhaps problem. It won't cost, that would be good.
Yeah.
That would be really good, because we all used to go when we were younger, well, me and Craig did anyway. We used to go on the boats with our dads, but it's nothing like that for a long time, so it used to be a great place where people went, but so few people go now, but they've got so many residents that live in that area that didn't before.
So it was something that was quite interesting to us, that since the residents and the population nearby had increased, the visitors had definitely decreased. There's more people in the area.
Craig always say,
which is-
Clatto residents.
We're finding it hard to entice people back into
Yeah.
most of Clatto. If we had the water
Free
attraction, that
beer.
would be the big,
Yeah.
big magnet.
Yeah.
Do you
Well,
think there's mileage in, because it is a public health issue, and there was a Tayside Health Board, blue-green algae working group, and they describe it as a public health issue. So you've got public health graduates here at the university. I think they could look at these guidelines as well, and look at the wording that's used, and look at the information that's available to everyday public. I've found stuff, I think I shared it with you, from Missouri, where it's a problem, but they're quite keen on the recreational use of water, and they just say, unusually for America, pay attention. And if you see thick green water, stay away from it.
I've heard that if you avoid the scum or the mat specifically, that's probably the best way to remain safe. I wonder what you would say.
In the Netherlands, I think it was somebody else was speaking somewhere at a conference. In the Netherlands, they say if you get in the water and you can see your feet and you're up here in the water, you're right. Personally, I mean, I'm surprised that they're going with the drinking water guidelines, and the WHO guidelines for that cell number is low risk.
Yes.
So,
In drinking water.
but for Clatto,
Yeah.
it's low risk, that cell number.
Yeah.
It seems like the media is kind of,
Well-
and it always does, with anything, sensationalised.
Sometimes in my more cynical moments, I think that it's about officer protection rather than public protection. And so that we identify such a possibly safe level that there's virtually no risk at all. That doesn't recognise the recreational use of the place.
The benefit of actually people using it and enjoying it.
Well,
they could walk into a tree or fall out of a tree or
I
something.
think crossing the road to get there is more dangerous than actually.
Yes,
Have
exactly.
you actually had any reports of illnesses recently from that?
Yes.
So
So
there is.
there's the swimmer's itch.
But that's
That's
nothing
a
to
common
do with
thing.
toxic.
And there's nothing to do with toxins. You're
And
quite
it could be other just
Yeah.
gram-negative bacteria in there that are
No,
not harmful.
anecdotally aware of people who've been ill.
Or
Two of the
hospitalised.
older members of my angling club, they're in their 80s, have just taken ill after fishing on an algae-infested water. They are blaming the algae, I'm blaming the pub lunch.
There is one aspect of, because it's such a huge subject that I did when I was talking about just generally blooms, the oral route of ingestion, there's quite a low toxicity compared to, I mean, all the data you see in the literature is usually based on mouse assays with an intraperitoneal, which is not really realistic. However, a word of caution, intranasal, so if you're windsurfing along and you have ingestion by inhalation, that is very similar with microcystins anyway to the intraperitoneal injection. So if you're sort of whizzing along in your boat or your windsurfer, there is an increased risk. And with the marine toxins, manatees, I don't know if you know what manatees are, because they come and just breathe at the surface, they're particularly vulnerable because they're just getting that waft of inhalation. So, that is another aspect of public health.
Having a look at just the cyanobacteria working group for the government, they list 30 to 40 reports of illnesses caused since 2002. But how many places are closed for years on end and not being used at all? So again, when we spoke about risk assessment, in this case, what's the risk and what is the damage caused by the regulations?
Well, one of Jeff Codd's early concerns was that what is the effect of long-term exposure to low levels of microresistance? Well, now we've got 30 years of that, and by 30 years we knew that smoking wasn't good for you, but 30 years on, there's not a strong link between.
Yeah, it's easier to work out. I mean, there is epidemiological data from China where there is increased liver cancer from people exposed on a regular basis to low levels of microresistance.
And they were drinking the water. That
They
was their
were-
water source.
Yes, that was their water source compared to people. But it's quite difficult these days. And I mean, smoking was probably quite an easy one to put two and two together. But with all the different things we're exposed to, is and lack of monitoring. Because I think one of the big issues is from this survey we've been doing as part of the costing. Some countries, they do have more strict regulations on monitoring, but in this country there certainly isn't. There's more of an avoidance. So the regulatory authorities only ever do it if they really have to. Water companies won't sort of say, oh yes, we'll do it and we'll tell you what the results are.
Yeah,
Because they don't, I've worked with them, they don't.
You don't want to spread that bad news. If
No,
there are
I think-
No,
I think
because
that-
then people get panicky.
I think, George, that's why you said about public health. Think about the wording of some of these signs and some of the information that's sent out, certainly the newspaper articles, and just assessing how appropriate some of those communications are.
Yeah.
And it'd be really interesting to see what people who specialise in that area would say on that, and we'll hopefully try and find
Yeah,
some.
yeah, definitely.
Hopefully have a look.
And just sorry, I'm just going to say a quick thing. I mean, 4 foot loch gets, I'm sorry, I can't say it very well. It's always getting a Phanes almina. That's really common in there because of all the bird poo and the occasional overspill of the sewage works in storm conditions. But that's never, to my knowledge, had any toxin producing species in there. I mean, the Phanes almina could be, but it's not morphologically. That has never been shown to produce. It's a different kind of one that does. But
One thing I was wondering, I know we haven't got much time, but just in terms of the water at Clatto, is there any flow through of water? So is it just, because that's critical, isn't it? I mean, I would imagine with 4 foot loch there is some flow, so there is a turnover of water in there, and maybe that helps to refresh it. Well, obviously it will help to refresh it to reduce the risk, whereas in Clatto, if you could really get some flow again, I mean, obviously it used to take water from Clatto Reservoir or somewhere. Is that pipe still operational? No?
No, they've closed it off now, so
Right.
that option's gone.
that's what they did in Australia. Said they had a drought, but they to have in the Murray Darling they have huge problems. So they did build the facility to add more flow, but then they got the drought, so they couldn't use it. But that was the solution.
Yeah.
What was the reason for closing it?
It was Scottish Water used to use it for topping up the main supply at Dundee at one point in time, but that's not required now. So Scottish Water have no interest in that reservoir whatsoever for use for the water supply for Dundee. So hence it's been cut off at both ends.
But if it still exists, isn't there a possibility that when there's plenty of water in Blackwater Reservoir, there could be a flushing through of
Yeah,
a lot?
they've tried that. The other difficulty is SEPA, because then you have to discharge that water somewhere,
Yeah,
and they're not terribly keen.
having algae
Yeah,
being washed out into the water.
Yeah. And so you need that kind of license, and you need that kind of agreement. So as a rain-fed source, the main thing that we need to do is ensure that surface water runoff that's got nutrient loading in it doesn't go in. That's a key thing. Beyond that, then find some method of treating that algae when it occurs. And also, using the measure that we've got at the moment, this 20,000 cell count, is an impossibly low level, and I think we need to do toxicity tests to see when it becomes toxic, to see if it's 50,000, 100,000, 200,000.
But they'll always be different.
It will be different.
It'd be interesting to do those tests anyway. Would you say it'd be worthwhile? Just you said that they're available for people to obtain. It'd be quite interesting to see.
Yeah. So yeah, it will vary, and it will vary from year to year and month to month. But I think as a contained water body, I think we can build up a profile of how the algae in Clatto behaves in relation to water temperature and climatic conditions, and
Sounds like a good student project.
get a good profile.
The
of
kits are designed to be used by anybody. I mean, we've done some work with a guy that has some golf courses in the US because they had a problem with their irrigation water and their grass was dying because it was in sort of shallow rooted sandy grass and sand, and we found he was using just a couple of different quickie tests, commercial ones, and then he sent us some samples for confirmation. But you know, they are designed for anybody to use with half a brain sort of thing.
Okay,
Yeah,
I think
no, I didn't mean. I didn't mean like that. I'm sure you very easily get automatic. No, it means anybody. You can easily delegate it to.
You don't need to be a scientist to kind of.
Yes, no. Just
Well,
follow a few simple cooking instructions.
We will need to stop there, so thanks a lot to everyone. Everyone's input is shown to be just what we were looking for, really valuable and really good angles, and that's really great, and it's really been helpful to us. We can obviously still continue to talk past this.
We
but
wish
we'll
you
need to-
all the best.
Yeah, lovely.
Yeah.
I'm really impressed with the work
Yeah.
you do, and we wish you all the best in the competition. So, well,
I'm good.